That's not an accommodation, that's talking about about like if one professor of non
Professor Rodriguez?
Yeah.
Was there a submission box for a research, for the third research paper?
that's.
I didn't load it till like
So.
the night, the day after it was due.
Okay, so can I just turn it in now? And I thought you
I
had
was sending
Google
you the
Doc.
Google Doc.
I normally go off the Google Docs.
She must
The
have.
other one, just so I can, I'll give you
Deadline
comments
meetings.
there, like just the grade, I'll put it in the tab, but I'm looking through your Google Docs.
Okay.
I'm running all the stuff through the ChatGPT stuff. So, you get an email about me asking you to put the honor code on top of your statement. That means that most likely my, the software told me that you have AI stuff. I'm sure you guys aren't using AI on your documents.
If I shared it with you on Google Docs, that's okay?
Yeah, I prefer, that's what I do, because that way I
instead of
have
uploading.
full access. Yeah.
Okay.
Like I said, in Brightspace, it says you can upload it there, but I'm just gonna put, it just gives me a nice column for me to put the grades. But again, stay away from AIs, careful with Grammarly, because it does have that generative writing or the fixing, and it's gonna push all your stuff to AI. Right? As you guys can tell, I'm not picky with spelling, I'm not picky with a little bit, if the grammar's a little bit off, I wanna see your stuff, so when you work with my stuff, turn off the Grammarly just to be safe, yeah? So be careful with it. All right, do your quiz, and then we'll get started.
Did you get a chance to look at the last question, the last quiz question that we talked about?
Yeah, we'll talk about it after you guys finish the quiz, right?
Okay. It's on Brightspace.
All right, good shit. Be quick, cause it closes in about like 4 minutes.
Closes at 12:10.
Okay, okay. Give me a second.
Yeah, it's a difference.
You got about a few more minutes, jump on Brightspace, do the quiz.
Google Docs or I changed the one because I just went over here. Yeah, that was 47. Oh, it is 6. But it starts at 4, so we'll see. For you.
Does it mean more time? Hour 30, yeah, pretty cool. The thing's gonna close
These are
at
so hard.
12:10, so do the quiz ASAP. All right, for everybody else, I think last week we had, no, 2 weeks ago or last week, we had the question about microtubules, right? So that was the quiz question. It was asking, how do the microtubules come together to create that filament? So half the class, like around half the class, picked the wrong answer for that one. So I want people who got that answer wrong to come up on the board and draw what the subunits of microtubule filament should look like. So if you got that question wrong, you guys come and draw it on the board what the microtubule should look like. If you guys do this, I'll give the points to the whole class back for that question.
The one from TV?
No, from, I think it was like.
Monday?
Monday, yeah. Not you. I'll push you for another one.
What was the question?
So microtubules, there was a question on Brightspace, and it was asking about what is the subunits that we use to build the microtubule filament, right? And then it has 2 subunits, an alpha and a beta, or how they arranged to build the cable. It was a little bit of a trick. Here we go. All right, we got one taker. Anybody else? We're gonna need another one. Let's do it. If you're still taking your quiz, don't look on this side. Yeah, this is other stuff. And then tell me what those spheres represent. Yeah, right there. We'll imagine that they're all the way to that. But how are they arranged?
You gotta remember, like, alpha, beta, alpha.
Yeah. Just, we're gonna have to adjust a little bit. So can you explain yours up here? So the individual subunit?
That's the beta tubulin is on the minus side because it gets hydrolyzed by the G2P to GUP. So the beta is the GUP side and then the alpha is the G2P side. And the alpha side is the side that's growing towards the outer part of the cell, and then the minus side is the part that's anchored to the nucleus.
Perfect, right? But in the question, it said something like, how does this cable get built? And it was like, there's dimers of alpha and there's dimers of beta that are alternating, right? And that would mean you have two alphas together, two betas together, and that's how you make the cable. But this is the right way, right? If you have a dimer set of both the beta and the alpha, this is what makes that individual subunit. Like you said, one of them is gonna be, they both are bound to GDP, but only one of them is gonna be, that beta subunit is gonna be getting hydrolyzed, right? And that's the one we're in. GTP bound, super stable, we have a strong cable. Once it turns to GDP, it changes the conformation and it's unstable. If it's inside the middle of the cable, we use this on here. GDP is in the middle of the cable. The fact that it's already built up will keep it stable. If the GDP ends up being at the very end of the cable, then that's gonna be unstable and this thing will start to open up and come out. So for yours, the only thing I would change is, yes, you end up having, like in the picture, it's alpha, beta, alpha, beta, alpha, beta. But when we think of a subunit, they kind of come in like this, alpha, beta, this. Yeah, so that's something we'll change this drawing, put it through the open circle. All right. And then this answers today's quiz, right? So everyone should have had it submitted already, which is, once we're building that cable, overall that cable we have what we call that positive end, and then we have that minus. If we're looking at a cell, and I draw this cable going from centrosome, and it's kind of going out like this, what charge would I have here, and what charge would I have here?
Negative, positive, two of them.
Right. So this is how it's arranged from the center out, right? Like that. I always forget which way the motor proteins go, so I'm gonna have to double check this. Yeah, kind goes to the positive one, dying is a negative thing, kind is positive, right? So we have a kinesin protein, and you grab on some microtubule, it'll move you from the center of the cell, it'll move you towards the outside, right? If you want to move something towards the inside, basically grab, you're gonna use a dyne, attach to whatever protein or vesicle you want to move into it, and then this thing will walk itself towards the minus direction. So kinesin moves you out, dyne moves you back in. Yeah, so I think that was the questions for today's stuff. What else? Yeah. This is a beautiful diagram. I really like this. You said one more thing, but you didn't add it here. That's the only thing I would add to it. You did mention it though. Right. Which is, this is the one that goes from GTP to GDP. Right. So you mentioned it, you know right now. You had given me this on the midterm, it would have been a beautiful answer. Yeah. Everyone feeling pretty good about microtubules? Yeah. All right. I think I'll give you guys the point just because it was a rough question. So I'll go in there and I'll try to change that quiz question. How do you guys feel about today's quiz? Good? Yeah. It's good.
Why is it only, it's doing like partials, but there were only 2 questions in terms of like, it wasn't like a 50 or 100, it was like point.
I got
Because it's a
1.6.
It's multi-select.
Oh.
So it's like,
Whoa.
in order to get this task done, you need to have multiple things. In
Oh.
Brightspace, you'll tell that they're multiple choice, it'll look like a circle when you pick it. If it wants you to do a multi-select, it'll make them look like squares. Like that.
Huh.
Yeah. So then, what are you doing?
There's a lot.
Oh.
Do I want to post it now? Maybe if you.
I mean,
You guys are, I'm gonna see if it's dirty.
all right, so multi-select, you had to combine all the things together, right? So in this case, what is the one thing you're gonna need for sure to help get that vesicle to move across these spots, right? I'm like always thinking about the center, and it's like the biggest foundation to getting vesicles to move we're gonna need the microtubules, right? That's gonna be our basically infrastructure needs. Based on what Paula said, if we're moving from the ER in the center to the Golgi, what order protein should we bring up? Kinesin, right, because we're going towards the positive side, we're moving out, so that's the next thing you're gonna do. To build the vesicle itself, what clathrin proteins are you gonna use, or sorry, what co-proteins are you gonna use to build that vesicle?
COP one, two, one
And I don't remember.
goes to the ER and two goes to the Golgi.
Yeah, so now at the beginning of the question, you're asking me, we're moving from the ER and we're moving ourselves out, so it should have been COP two, right? If you wanna move it backwards, then you have to switch over, you use COP one, right? And then we switch over to this guy, we have to switch over to using the kinesin to basically move us back. Yeah? Everyone's feeling pretty good about this? Like I said, it's a beautiful question to put on a midterm, guys, especially to have you guys explain stuff. I'm gonna have fun with it. Again, beautiful drawing, thank you. All right, so now let's jump into today's stuff. Before that, I messed up on the syllabus, I was looking at it, I skipped the actin lecture. So we're supposed to go microtubules, then actin, and then we bring up the vesicles, but I completely left them out, I skipped the slides. So now we're gonna have to go back a little bit, talking about how the cables are made, right? So we're gonna go a little into greater detail about how Actin works today. With that being said, we need to talk a little bit about scheduling. So I've been shuffling the schedule around a little bit. Right now, what I'm thinking of is I pushed the last set of lectures, I'm pushing them up. I'm hoping to get all the lectures done by the 24th, right? Then for there, sorry, on the 22nd, on the 24th, I'm gonna send out a paper, but again, this is the one where I'm gonna give you extra feedback, I'm gonna give you guys a price-based announcement, but I want us, as a whole class, we're only gonna pick one paper. And we're gonna behave like this was my other class, which is 243, where I really want you guys to appreciate the molecular techniques that they have behind, like that specific project, trying to understand the molecular aspect, but we'll focus on the techniques. For that, I'll tell you kind of the things that I want, understanding the methods. It's like you have to understand the principle behind it, and then it's like how we can get a measurement from it, or how we can get an idea of how things work from it. We'll come into class, I kind of want to discuss it as a group, and I think that's what we'll do this day. So the paper should already, I'll give it to you guys, hopefully tonight, you guys can start reading your paper, but on this day, you need to come like where you have already read the paper yourself. Yeah. If you have questions, it'd be great. If you can answer stuff, I'll be giving out extra points for that. Yeah. After that, April 26th is the scholarship Sewanee, right? So there's gonna be presentations going on all day, and there's gonna be poster sessions in the afternoon. We had talked about doing an extra credit assignment, right? Like a video project where you could basically show something and then afterwards make a little side video showing about how you connect it to the work in the class. It's a really good place to go pick up ideas about how Sewanee is using molecular biology or science in the school, right? Like the research that's going on. So I was thinking of canceling class that day just so you guys can all go, pick a talk, pick a poster session, go check it out. And that way, you can make your video based on the topics you pick up there, yeah. Then after that, we'll do the last, time it all together, where it's like, I come in, I rush through the PowerPoint slides, and the following day, we'll do the final review where you guys ask me questions about what do you want to go.
So is this the video extra credit, is this the same as the other one you were talking about where you have to write like 2 pages, or are these 2 separate like assignments?
So I've been thinking about that, because I'm like 25 points is a lot, especially for a 2-page assignment, it feels unfair. So that's what I'm kind of thinking, I'm like, I almost want to push all of you guys to do the video thing, right? Because, or otherwise I have to bring it down and just make it like maybe 10 or 15 points. Because the videos take a little bit more time. And the best part that I'm liking about it is that you have to put your ideas and your understanding into the video. The text, I'm a little sensitive now with AI stuff, where I'm like, I don't want it to be contaminated, yeah? So I'm hoping I'll put it on my to-do list when I get back. I'll give you guys more comments on the AI video, how I want, like ideas on how to write it or guidelines, how I'm gonna grade it. And then I'll give you the paper and I'll give you a little bit of how I want you guys to break it down.
So if we do like the video project, then it would still be like the full 25 if we did the full video project?
Yeah, that's the goal.
Or, you know, or more if
I
we do
because
like
I'm
really
trying to
good.
push myself with the grading, because I need it, just to see like it can help bring the grade, the class average up, right? But I'm not too sure how much of a shift it'll cause. And, but let me finish the grading, I'm rushing it, you guys, the next few days, I'm just gonna be knocking it out, and then I'll give you more info on it. Yeah, so that's the plan, everything pretty good. The final, I think it's gonna be in class, guys. I've had a little bit of, again, contamination stuff, which I don't like, so the cool thing is, it's the questions, because it's not open notes, I'm gonna have to go a little bit more on the surface, so I need you guys to understand the big ideas. Even if you don't remember the keyword, argue it out as best you can, yeah?
The final just covers unit 4, correct?
Correct, it's
Okay.
not cumulative. I don't wanna lose you guys and just be like, hey, I'm gonna drop out, stop paying attention. So remember I told you guys you gotta get me at least above a D and I'll do the grade dropping, right? So I need you guys on your toes to keep going, yeah? So let's say you had 3 A's already and you're like, oh, I'm not gonna take the final. It's like, no, you gotta take it and at least give me a D.
You said it's not open note or anything?
No, it's not gonna open up. But again, I'll change the questions because of it. Yeah. And you'll be able to argue more about how the systems work. And this is probably the stuff I'm gonna bring up to you guys, yeah.
And even though it's like on a final day, so we would have like a full 2-hour period to do it, is it gonna take us the full 2 hours to do it, or is it gonna be structured like a normal
It has to
50-minute
be like
class?
the first midterm, because they're the same. If it was worth more points, then yeah, I could go bigger, but no, it's gonna be closer to midterm 1.
Okay.
All right. Pretty good? On the page, all right, awesome. You guys spread the rumor about me that I hate AI, so if anybody else asks you to take my class, be like, watch out, that guy. He gets mad about AI stuff that way. They don't think of doing it, yeah? So help me out. If your friends ask you, be like, yeah, it's cool, but watch out, if you use AI, he chases it down hard and chases the whole class, yeah? I want that said about me, that way the AI people don't come to me. All right, let's do it, guys. So Actin, sorry about this, I messed up on the slides in the order, but now we're talking about back to the cytoskeleton, about that mesh that we have that's gonna help us build what should our cell look like, and it can affect how the compartments behave. So for this, we're gonna play with Actin. We're gonna connect it really nicely with this cell motility migration. I'm gonna emphasize one here. We'll go a little bit into how it can help with the vesicles and trafficking. And then I'll show you down here how we can actually use this to close off the cells after cell division. And then this is a really awesome thing where we have these super specialized cells that, even though Actin has their own little motor proteins and they're very weak, if we add them up, they're weak and small, but if we add up their behavior, like that's literally how our muscles move. So it's like something super small but additive can build something really powerful. Right. So for this one, it's just a little bit different from microtubules. So one of the things is it has, like this is basically would be the subunit, right? When it's by itself, we call it globular actin or G-actin. So that's a single subunit. When they glue onto themselves, it starts to basically start attaching to themselves. It's a concentration dynamic. If you have very little, when you build a dimer, it likely falls apart and it will go back to just being G-actin, right? Like your thing will stay in that state. If you have a lot of G-actin present, then it favors that once you get like these small amounts of them to come together, and you can call it filamentous action, they'll build this small thing called like a nucleus. So that'll be the base of where this thing is going to keep reacting. So if you keep high concentrations of the actin, then they'll keep adding, and you have growth at both the plus end and the minus end. So that's a little bit different from the microtubules, right? Microtubules, they fall apart at the minus end. You can't build them from the backside. All right, so yeah, so single subunit, this builds a chain, and you can see it's basically a set of alternating, looks like it feels like it's alternating too. All right, so like everything, this is another similarity to tubulin. I want you to be thinking about, in order to make a filament be able to polymerize and fall apart, you have to change its conformation, right? In order to do that, you need to power up the reaction. For our tubulin, this is where the power is coming from our GTP to GDP. When we look at actin, we're getting that power going from that ATP to the ATP actin. Yeah, of course, give me a second, guys. Yeah, my bad. My bad, I might rest a little bit. Let's go back. So we're starting to build the cable up. Remember, the hard thing is, it's hard to get this thing to build up, to build that base cable. Once you have a base cable, it's easier to keep doing stuff because you have a stable thing to attach it to. In this case, we have different proteins that are gonna help build this cable and different proteins that are gonna help break the cable, just like we have with tubulin, right? So for this one, we can see that basically this formin is kind of helping function as almost like a base, right? So it's hard to build that small nucleus of those first big on, but if we use formin, that could be the base where we can start gluing the ATP actin onto it. So that's one way that if we control where this is present, we can determine which molecule, when we're going to start building the actin cable in a specific spot or in a specific cell type. Yeah. As we do that, it's going to get helped by profilin, which is going to be the one in purple, and that one, it acts like a guess, so it's going to be switching, but exchange factors. That's the big key thing. We're going to be switching from I wrote it backwards. Yeah, you're kicking out an ADP and you're bringing in a brand new ATP, right? And that's the way that you're basically putting this in the high energy state. This is that switch of, now this is the ATP conformation. We've invested the energy, and that's what's going to give us that change in conformation where actin now is able to build. All right, so we started building the cable. Now we're going to get it to keep going, right? Sorry. I mixed these guys up, my bad. We've built the cable, and we have a cable that's already set up. Actin has a really cool thing where, again, remember, it's like, it can be growing at the plus end, it can grow at the minus end. It's based on what you have in that environment. On top of that, you have, again, more of these molecules that can really help us regulate how it works. R23 is one of my favorites. It's super interesting because what it does is it attaches onto a growing cable, and then it actually helps create like an additional branching point. So you start with an additional actin, R23 comes in, and it'll give you a little side branch. And this one is super important when it comes to cell movement, and it has a really cool mechanism of how it can expand the cell membrane. We're going to go into it a little bit later today. So R23, once you remember branching, and there's going to be other proteins that can further stabilize this interaction, kind of like these, where it's like once you set up the fibers and they cross each other, you can have 3 actin binding proteins, and they interact with themselves and actually will hold the cable together. So you can make a mesh with the R23, and you have additional proteins that come in and stabilize that mesh, or you can stabilize bundles, right? It doesn't need additional proteins helping you. And finally, it's the thing I wanted to get to. If we zoom in and see how actin itself is behaving, right? This, you should see a lot of similarities to what's going on with tubulin, right? Which is, when we're adding the molecule, it's coming in with that ATP, right? That's that conformation where it's like we've invested energy. It's in the conformation where it wants to build filaments. Once it's in the filament, it hydrolyzes the phosphate group. Now we have that ADP. In that case, it's now going to be, let's say, that unstable form, the one that wants to fall apart. This means that the minus end, where we have a lot of the hydrolysis happening, we're going to have more things falling off, but you can still get a few of ADP to actually jump back on, because they're just basically kind of gluing themselves directly into each other. Instead of this one, which is in the back end here, it's a whole straw that you're meeting with, right? Like you have to glue them, they have to plug in to their left neighbor, they have to plug in above, and a little bit of their cap to the right. Trying to fit into that pocket is so hard, versus this, which is 1-to-1, it's easy to move them back in. So that's a little bit different about these guys. But the cool thing or dynamics about this is that once you have them fall off, we sit this thing in the ADP. This could potentially go back into this region because it'll be stable there. But in order to recycle these, we basically have our GEF function, right? But instead instead of switching GTP to GDP for GTP, we're working with that exchange of brand new ATPs coming in, and we're kicking off that old ADP, right? So you guys know I love that kind of question. Like, hey, how did it get activated? Where's the phosphate coming from? In this case, remember, it's a brand new molecule, whole GTP with 3 phosphates, and you're kicking something off with 2 phosphates. There's no independence. Now that if you reset this, this is that happy conformation, or that conformation you invested energy into it to refold its conformation. Now it can come in and you can build it at the front end, right? So this is really cool because what you can do is you can change the dynamics of this molecule by controlling the concentration of the actin subunits, right? You can disable other cables, you'll have a bunch more actin in these. You can also control the building at this level. This function of swapping out ATPs to ADPs, if you control it, you can make an enrichment of ATP versions, which means we're in a building phase, or if you stop it, you'll basically stick at this side where you want to be building it. You're gonna make it unstable. Yeah, so super powerful tool. With this process of like the cables, you can just like, if the cell's moving forward, it literally dissolves the back end. There's a cytoplasmic flow where they move up to the front, and in the front, you ATP exchange, and then you can build more cables. And then the old cables dissolve, they shift forward, and you can keep building, moving in a certain direction. So it looks pretty cool. Again, playing with more of the dynamics of how this cable is going to behave, I want you to keep drawing analogies to what we're thinking about the tubulin, right? Which was, it has by itself its own little dynamics, but we can enhance those with different proteins. So we can put caps at the plus end, we can put caps at the minus end. At the minus end, it's where it's normally falling apart really fast. So by putting a cap at the minus end, you're going to stop that. It'll stabilize your cable, right? Opposite to that, if you put a cap on the plus end, right? What are we doing at the plus end? I heard building, right? So you're building it. If you put a cap on the plus end, you're basically going to stop the polymerization. And if there's nothing on this back end, you're basically going to dissolve that polymer, like your cable. The back ends will just fall apart because that's what they do faster. Other ones, as you can see, like this triple lysine, they can interact with like the middle of the cable, and again, it'll kind of help stabilize your molecule, right? And then opposite to that, you have this one, coffin. This was pretty cool because you can see it binds onto the actin and it actually trims them in half, right? What this does, it opens up 2 things where it's like, you have a brand new plus end where you're trying to build things, and you've created a new place where you have a minus and it can get dissolved, right? If before this cable was necessary for something, well, we needed to have it around all the time, we put a cap on both sides, and you have a permanent cable, right? It can't fall apart. In order to control that, that's when you bring in the coffin, right? Coffin is going to come in when it's time to get rid of it. You cut it down the middle, and now you have 2 open ends that can have different dynamics. Either build or you collapse them. And then, again, one more analogy, right? In microtubules, we talked about kinesin going to the plus end, kinesin getting us backwards, right, towards the minus end. For actin, they have their own set of proteins. They have the myosins. Most of the myosins, where is it? Yeah, so most of the myosins go towards the plus end. Only myosin 6 goes the other way, towards the myosin. So for the quiz on this one, I'm gonna basically keep it at myosin for the general, it moves towards the plus end, right? This will be our protein. And the way this one does, it's a little bit different than microtubules. Because remember, in microtubules, these proteins have, like I said in my head, I remember they basically have like feet. The blue one, like acid hooks on microtubule, they play with the dynamics of the back one, they bring it forward, then now that one attaches and it releases. This guy, they only have one. All right, so they have a single foot. And what we're doing is as you change the dynamics of that foot between the ATP state, hydrolyzing the phosphate group, and then releasing the phosphate in ADP, we're getting it to move forward across that actin cable, right? So it'll basically, they call it, I think it's my next slide. No, I think in the book, or maybe it was here, they call it like a, it's, I think it's like a cock phase, like it's ready to pop and then it pops, grabs, and it just basically does kind of this inchworming where it grabs on and pulls itself forward. But in order to actually get movements, what we have to do is you have to kind of combine this, and you normally have them in big groups. So there's a bunch of myosin basically working together, right? You know, here what we're looking at is we're basically zoomed in into like a part of like our muscles, like a muscle fiber. And the way that it works is here in yellow, that's basically our actin, right? We have the cable, you can see that we have it wrapped around, so we have stabilized it. And then this will be the base of what we're gonna pull in that direction. Same thing at the other side. Another actin cable, you have your proteins that are stabilizing the actin filament, you don't want it to fall apart, and it's anchored to this side. So now, if we think about it, it'll be like, if I was the actin filament, I'm sitting on this side and I kind of have my arm stretched out. She's on that side, you'll be actin and she'll be actin, right? They're attached and they have their arms kind of hanging out like that to make the cables. Now what myosin does is you have this cluster and you have almost two groups aimed in different orientations. When you have that, you're gonna have this huge cluster. Each one of this is gonna be one of those foots that does that job where it grabs it, pulls it a little bit forward, and then lets go, stretches, grabs it again, pulls it towards itself, right? When you activate the system, what's gonna happen is the ones that are aiming towards, let's say, it'll be this side, right? So towards Lindsay, they're trying to walk towards the positive end. Like they're basically putting on her arm. Then on the other side, it'd be the other ones would be like grabbing an hand, right? And they're walking, they're kind of putting on hand. So as you pull both of them together, this thing doesn't shrink and these don't expand. So what ends up happening is the base they're anchored to, this green part's gonna move this way. In this case, the green part will move this way. So in my analogy, if both of them have their hands up and I just pull them towards each other, their arm length doesn't change. The only thing that would change is where they're sitting, right? I would shift her this way, and I would shift her that way. Does that make sense? Yeah. All right. So yeah, you have a lot of these working together, and this action is literally the thing that when you guys are like flexing your muscles, that's literally what's happening inside. You have a bunch of myosin walking across actin to pull your muscles together. And then for cell division, it's you have the actin builds like a ring on the inside of your cell. So in the cytoplasm, you get that actin ring. So it'll be like a lot of cables. And what you're doing is, as you're walking across those cables, you'll actually start pulling them towards each other. Right? Same thing like we did my analogy with them. Imagine she was a cable strung around this side of the cell, she was a cable pulled around that side. As I pull them towards each other, they're gonna make a smaller and smaller radius, right? If I've been pulling across the top, there'll be another molecule at the bottom pushing across the bottom, making that actin smaller and smaller, and that's so we can squeeze that cell. For this one, I'm not gonna go that heavy into it because I prefer this one because it has more information. But again, another similarity to microtubules is we have that cable. There we have the myosin head, which can help us with the movement. But again, because now we need to have it, it only does it by itself. You kind of have to anchor it to another one so that you can get that thing to walk. And here we're only doing the walking towards the plus end, and we're burning ATP to get this to happen. But we also have to anchor this to whatever we want to carry or whatever we want to move. So this means that, again, if you want to bring in a vesicle, we have to attach ourselves to the membrane pointers of the vesicle, and then we can drag them forward. We can also work with the intermediate filament. This is the one I don't like. I normally don't like talking about it because I don't like it, but Remember, this is the filament that's built up around your nucleus, around your ER to help give them the shape. So this thing, when we disassemble that filament, this will be the thing that you can kind of use to basically move these things around or shift those different cables around. All right, so like I said before, this thing is super small. We're burning ETP every time you do it. But all these really small behaviors are adding up to do something powerful, right? Like I see most of you guys at the gym, you guys start lifting those big weights, and it's like you're doing that literally with little strands of cables and a little bit of ATP. So we zoom into one of the cells. This would be like a representation of the little muscle fibers that you have. This would be kind of look at a sarcomere. So again, this lead disc would be basically where they're attached or like the anchors. In that case, it's the analogy back to its pan and its lineage, right? This yellow part would basically be their actin, so that would be like their arms they're basically just holding out like this. And then the Z band or that center part, that's where you're getting the the A band. That's where you're getting that myosin to do its job, right? It has two arms. When I work on these two arms are working in opposite directions, and it's gonna pull on both of them together. So as you imagine, when you whatever links you have here, relative to here, it's like this is like a third, maybe a quarter. This thing will be able to move a quarter this way, and that will move a quarter that way. That movement is going to be able to bring, like once this thing goes and sets off, these two bands are right here. This is going to get compressed all the way to there. Right? So that's how your muscle is. It's long right now, and then when you're trying to flex it, you're basically bringing all these things, like each one of these little bands, you're taking away this little yellow area, right? And you're compressing it away, and each motor protein, each myosin that's grabbing on, we're burning ATP to get this thing to happen. And it'll be like, move forward, burn ATP. While this one's still grabbed on, another one does its process. Let's go, grabs on further ahead, burns off this ATP, and then you just keep doing this, right? And that's how they can kind of grip something and put it all together. But super powerful stuff. All right. So this is the one that I'm going to dive into. And I really want you guys to feel comfortable with the analogies of and Lindsay's arms, right? Which is when we're getting these things to work, it relies on the fact that these actin filaments have to be stable, right? So in order to achieve that, look, we have our cap proteins that are going to be protecting the ends. We also have the protein that's kind of wrapped around the actin filament to keep it stable. If you had an error with these cap proteins and your microtubules, basically, your actin breaks down, you would like your muscle wouldn't be able to the myosin would be able to grab onto itself, and you probably rip this cell open. Because right now it has a lot of stress, or not stress. It has a lot of mechanical force because it's ripped onto those. Let's say, and this is somebody's arm. On the myosin, this is the actin filament. It's like there's a bunch of the myosin that are grabbed onto this, holding onto it, like attached to it. If this thing falls apart, you basically disconnect your muscles. Like, I'm not sure exactly how muscle tears work, but if you rip them at this point, you're pretty much gonna have a really, really bad day. So for this one, right, we don't want actin change. I want you guys to feel comfortable with the plus and minus end, where we have it currently for the filaments, how they're stabilized, knowing that they're anchored to this. This one normally doesn't move. What's gonna move is these are gonna move relative to that, right? Like they're gonna be getting pulled in. Yeah. And then the other one is, yeah, the actin filament doesn't stretch, or it's basically a fixed length, and we're just walking our way through it to get it to compress. Let's see what else. All right, so in this case, right? So in the GDP state, right, it's like we burn the energy to get this thing to release, but when there is no ATP, this thing's gonna be down and it's gonna be basically attached to it. So it's like if you're holding it, like holding your muscle and it's tight, or firmly gripped to a spot, they should be grabbed on. You're gonna start burning that energy, and once you release it, you're gonna do that confirmation motion, right? So, there's gonna be ATP does the release, change confirmation, and then the hydrolysis changes the confirmation one more time, right? So that's your, I think it's what they call a cock, and that's your spring phase. It's like it's pulled back, and then basically cocks forward. As you're doing this, you're gonna be, when you release again, the ADP and the phosphate group, that's what ends up giving you basically a little bit of the movement that's going on with your molecule. All right. There is copper information. Yeah, and then you can see this is the last part where you basically release the ADP, and that'll be that last reconfirmation of change. You basically said it, it's folded itself forward again, and you have to do the cycle one more time. Bring in a fresh ATP. Bring in a fresh ATP, get the thing to release. It's gonna grab on, it'll bend itself forward, and just keep doing this little thing like that, inching itself forward. All right. And then the other one is, remember, it's like when you're working out, sometimes like you might get dehydrated or you guys get cramps, right? And normally the way you fix it is like they're telling you like you're low on salts or ions, right? So most people drink like Gatorade, or they, like I remember some of the guys at our school, they used to drink pickle juice to try to bring up their ion, like right before the games. And it's because the actin filament itself has a little bit extra regulation. And that's where this complex is gonna come in. And the big thing there is that it's basically gonna affect this little binding pocket that's currently on actin. When calcium is not present, this protein complex is sitting and it's blocking that myosin binding site. Right? So in order to get the cable to be accessible, you gotta bring in the calcium. Calcium reshapes, like you gotta change this protein complex, but the key thing is you open up that myosin binding spot. So that means that if you don't have any calcium, this protein is gonna hold your actin filament kind of trapped. Right? So no calcium, you can't grab your myosin protein, doesn't want to do the work. You need to bring in a lot of calcium to get them all to shift. And now, let's say it was a ladder, you open up the ladder and you can now use the rungs. Yeah. So for these two, go back, chase down how the calcium is going to help us, and also how we're going to use a lot of ATP to get this process to happen. Right? We use ATP to build the filament, and then we have to use ATPs to get the myosin to work. Any questions? No? All right. Awesome, guys. All right, you got my fire.
Extra credit video, does it have to be over like a specific topic that we see?